My name is Mariana Softman, and I'm a graduate student jointly advised by Linda Griffith and Paula Hammond. And today I'll be telling you about our efforts to create a fundamental tool to standardize stem cell production in order to make stem cell therapy a reality. And we're doing this by developing a synthetic polymer substrate upon which we can grow and expand neural stem cell populations that are uniform and homogeneous in their characterization and efficacy. So we're all familiar with our increased life expectancy that is going to happen in the future, as well as our growing population. And this will be met with a rising incidence of neurodegenerative diseases. And in particular, Alzheimer's disease is one such disease that is expected to grow by the year 2050 in the elderly US population. And this will be met with devastating personal impacts as well as economic consequences. Because of this increased concern, there has been a rising increase in investment from NIH, for example, in order to understand and develop therapies for treating neurodegenerative diseases, such as Alzheimer's and other related dementias. And there's also an increase in funding in the private sector. However, despite this increase in investment, a lot of pharmaceutical companies have tried and failed to produce traditional drug-based therapies. This is just a subset of some headlines from pharma companies that have not been able to produce an efficacious drug for Alzheimer's and Parkinson's and other related diseases. However, regenerative medicine and cell-based therapies have emerged as a promising direction for treating and addressing these concerns in developing therapies for neurodegenerative diseases. And this involves the strategic placement of cells or cellular material at the disease location, and upon integration, the hope is to find a treatment for these neurodegenerative diseases. There's been a steadily growing number of registered clinical trials for these cell-based therapies, in particular stem cell therapies. And this market is expected to grow. The global stem cell therapy market is expected to reach $145.8 million by the year 2021, and it's growing at a compound annual growth rate of 11% during this period. Consistency is important in any industry, and this involves consistency in the reagents, materials, and processes used in the development of certain therapies. And this is especially important in the stem cell therapy field. It is important to maintain a consistent source of stem cells throughout the long roadmap period for the development of certain therapies, of these therapies. And this includes from initial bench-side research, where the cells are being characterized at the bench, or up through the 10-year process of eventual integration into human trials and clinical therapies. Every step of the way, it's important to make sure that the source of stem cells is homogeneous and uniform in their characteristics in order to ensure a consistent and efficacious response during the therapy. And we're interested in tackling this problem. There are many standardized and established protocols for culturing stem cells, and the predominant substrate that is used for growing stem cells is called matrigel. So matrigel is sort of a witch's brew of a material. It is a gelatinous protein that's secreted by mouse tumor cells, and it's highly enriched in proteins that are native to the cell's environment, and it provides an optimal growth environment for the stem cells. However, there are some concerns and issues with using matrigel, especially in a commercial setting. So the inconsistency in the matrigel itself results in batch-to-batch variability. Additionally, because it's derived from a tumor cell line, it's not ideal when eventually being translated, because this can result in contamination in the cell population, which will result in tumor formation potentially. Additionally, any animal-derived product poses an inherent immunogenic risk. So with these concerns pose a risk of producing a stem cell population that is not fully differentiated into the type of cell that is desired, or a population that is mixed in its cell type. And these can ultimately result in a tumor formation or an adverse immune response when ultimately translated into a therapy. We're interested in tackling this issue, and our approach is to use a synthetic polymer instead of this mouse tumor cell-derived cell culture substrate. And the synthetic polymer can be used to culture and expand the stem cells. So we're all familiar with polymers, and the type of polymer we're using is kind of a glorified jello in a way. So it starts with a polymer backbone that is interconnected with a crosslinker in order to create a water-swollen network. And the crosslinking chemistry can be fine-tuned in order to change the stiffness of the environment that is suited to the cell's needs. Additionally, we can incorporate bioactive modifications that are native to the cell's environment in order to further provide a physiologically relevant growth environment for the cells. The advantage of the synthetic polymer platform is that it's not animal cell derived, and there's a low risk of having a tumor formation response or an adverse immune response. And additionally, it's consistent in the formulation and also reproducible. So this addresses the issues with Matrigel currently used. Importantly, we can fine-tune the polymer chemistry in order to design and control the differentiation of neural stem cells into different lineages. We're ultimately interested in developing a protocol for producing the large-scale expansion of these neural stem cells into their different lineages that can ultimately be purchased by clinicians, researchers, and partnering industries. And we're ultimately interested in providing a fundamental tool for that can be used in the growing field of stem cell therapy market. I'm fortunate to work with a very talented group of scientists with a very diverse set of background in polymer chemistry, stem cell biology, as well as fabrication. And I'm happy to answer any questions at my poster, and I'd like to thank the Deshpande Center for providing this opportunity, as well as my catalysts, Bob Farrett and Bonnie Fendrock. Thank you.